Yeah, so Fred Hutch in particular has a phenomenal prostate cancer program. So that we have our lab in particular has some strong collaborations there with some of the clinician scientists that are really involved in seeing the patients and then thinking
Everyone, all
about
backgrounds, all
the
ages, all
research.
First of
genders, and all
all,
walks of life
Patrick,
deserve
thank
an
you for joining us
education.
today.
Come be one.
This is super exciting to have you on. Helen was asking me what kind of topics I'd be really interested in, and I saw that there was the opportunity to talk about biomarkers, about cell-free tumor DNA, about bioinformatics, about from with a postdoctorate researcher from Fred Hutch Cancer Center, and his name happened to be Dr. Patrick. I don't know, it's coincidental, but guys, Dr. Patrick is in fact joining us today, all the way from the Fred Hutch Cancer Research Center, volunteering his time to discuss with us his research. Thank you so very much for coming on, Patrick. How are you doing today?
Great. Yes, thanks for having me and for all the work you're doing here. This is awesome to be part of.
It's a joy to have you. We've actually raised 1,600, a little over $1,600. We have two times
Nice.
donation matching on, so every dollar counts twice. And yeah, it's all going towards research that, for example, you and other members of Fred Hutch are doing. So, yes, Patrick, if you wouldn't mind giving us a little primer into who you are and what got you into science. I always find this to be a really cool question because everyone's journey is just so different. What was it that got you into the science side of things?
Yeah, so I am in computational biology at Fred Hutch, which can mean a lot of different things depending on what lab you're in. But our lab is very much focused kind of on the computational side. I would say cancer genomics, data science blend. So we'll hit on, I think, a lot of what you mentioned in that intro kind of throughout our conversation. My background personally is more in mathematics and statistics. I've kind of always had these interest as well in the biomedical sciences and the public health sciences. But even though you're calling me Dr. Patrick, I was never going to be a medical doctor. I knew that. So kind of this route into computational biology was a way to like leverage that math background and the nerdiness around data into to doing something meaningful in cancer research. So it's been kind of some twists and turns along the way. I thought I was going to teach for a while and then pivoted midway through and then kind of fell down this rabbit hole and still kind of question how I actually got here and get to do the really cool work and I think impactful work that I'm doing now. But it's been a really cool opportunity and excited to keep forging ahead.
What was the thing that motivated you into liking math? So, right, that's something that we always hear about is that, oh, I can't do science because it has so much math in it, or math is so hard, or I don't like math. But you actually have turned it into a full-on, like, research driving element. What, what, if anything, were you connected to math? Like, was it an early age kind of thing? Was it you found programming? You're like, this is my jam. Just for folks at home, like wondering, like, if you want to integrate math, what would be the direction or potential direction you can go to be able to do that? And I guess what also made you not fear math?
Yeah. So some of the family that's visiting currently is my mother, who is a high school math teacher or was for many years. She's recently retired. So I think that has always been a huge driving force. I mean, I kind of grew up going to the high school and hanging out with the math teachers, and they would come over, for different afternoons or evenings when I was growing up. So that was always math was a fun environment and something I kind of just gravitated towards naturally. But I mean, I enjoyed like science classes and other subjects. But yeah, I mean, I was on the math team in high school, so yeah. And I thought I was going to teach with that because that's what I was exposed to growing up. And I do have a passion for education and some of these type of like science outreach initiatives. Um, or a lot of people go into finance with mathematics, which I always found very dry, and so I always thought there had to be something different I could do with that kind of married some of these other interests I had, and that's kind of like through talking with other people, kind of through my undergraduate career, I found biostatistics and did some research experiences, and that's kind of what's led to eventually going down this route.
Did you, when did you discover that you had a love of some of research of the more this hands-on like in biomedical element to it? Because like you were saying, it's a lot of the journey is the statistics side of things and the finances, and to go towards the research route, I imagine there had to be something that kind of grabbed you that was like, hey, this is something from a biological puzzle perspective that is really interesting. What was that thing that kind of drove you towards where you are today in terms of working with, cancer, right? Like you said, instead of something like finances.
Yeah, I mean, I feel like cancer is something that impacts all of us in many different forms. Whether it's personally or a connection with a friend or a family member. So like that's obviously something that's always just very grounding in the work. But I think for me, with mathematics or statistics, some of it can be very very theoretical and can feel very far from being something that's like actually implemented and feels useful. And so I've always gravitated to more the applied side of things. And so I always wanted kind of the research I was doing, or like the methods I would be developing to actually to make a difference, to be used, not just to live in some academic journal, just to be seen by people in statistics. Like that has never felt valuable to me. And so I think with the work I'm doing now and kind of where I've steered things, like there is like clear direct impact. We're working with patient data and trying to make impact on how we think about cancer and how we can potentially develop new treatments and potentially change treatment regimens and adapt things on the fly. And that has like very like you go and you talk to patients, and that's just always a really impactful experience. And so that's what I think I value the most there.
Nice. Now, I guess let's jump into it. What is your overarching research question? Like, what is it that you're trying to develop a tool for? What is the big thing you're trying to answer? And then, of course, the evergreen hitting, why does this matter? Why this is important that we should care about beyond just, right? I mean, there's a connection for human health, right? But just even from a basic science perspective of why this is important to know.
Yeah. So our lab and kind of where my research was before coming to Fred Hutch was based in this liquid biopsy. And so I think we're all kind of familiar. You might be familiar with your traditional tumor tissue biopsy. And so if someone gets they catch a tumor on a scan, what you're going to do is probably go in there and try to get a piece of that tumor tissue and do some type of genomic profiling, and you can kind of do your cancer staging, and that really drives kind of treatment options that patients are then going to undergo. But that's very invasive, and that's really only a small snapshot in time. And there's also some tumors that we just can't even get to surgically or with a needle. So kind of this liquid biopsy approach has emerged kind of in contrast or as like a complementary approach to that, where you can take a simple blood draw, or you can use other bodily fluids like urine, or there's like fluid from the lung that you can extract as well to get some of your lung cancer information from. And with that, there's a whole host of different biomarkers you can look at. And we're all familiar with getting our standard lab panel. Hopefully, we're going to see our primary care and getting a workup. So you have those type of biomarkers, your enzymes and things like that. But also, as your cells turn over, and this happens in completely healthy tissues, not cancer related, they kind of release their DNA into the bloodstream. Much of that gets degraded and just kind of gets chopped up. But some of that you can actually detect in a standard blood draw. And then so with that, in cancer patients, we also detect DNA fragments directly from the tumor. And so that's a way that we can kind of try to capture information directly about that individual patient's tumor over time while they're on treatment, or maybe in between after surgery, and monitoring to see if there's any signs of residual disease or throughout the treatment time course. And you can see how the tumor is evolving and responding to different treatment pressures. So it has a whole host of different applications in the cancer context and also in other disease contexts as well. And so our lab is really focused on kind of developing the methodology to be able to kind of computationally extract the different information from those liquid biopsy samples.
And so I guess one really big question is how can you tell where the DNA is coming from, right? Because it seemed, a tumor is heterogeneous in terms of the DNA mutations, right? So that tumor mass is not just one type of cell, it's not one type of DNA mutation that makes that tumor the tumor, but it's a whole mass of things. How do you know what you're looking for in that tumor in terms of the identifier that this is in fact tumor DNA without comparing it to regular healthy DNA? Is that a per patient kind of thing? Is there a universal marker? Is there both? Lead us through the logic of how you would parse those two elements out.
Yeah. So I mean, so we're most of us are familiar with kind of the standard, four bases of your DNA sequence. I mean, that's going to be shared by all healthy cells. And then with cancer, there's mutations there, and some of that sequence changes. So those are some of the things we're looking for. But there's a lot of different kind of epigenetic features that turn things on and off, and a lot of this can help us. Pick out different cell types or tissue types that these fragments are coming from, then also distinguish what is tumor versus what is just a healthy circulating cell-free DNA fragment. But particularly, we're often looking for, um, like you said, different mutations that are different from the healthy normal. And so a lot of our samples, you can tease apart like a healthy baseline and compare it to the tumor sample, and then you can tease apart and figure out which are the tumor-derived fragment. And then we're looking at different types of mutations, different types of structural alterations where large chunks can be rearranged across the genome. So that's where a lot of the computational tools come in into developing that, where you're doing kind of the bioinformatics and the sequence processing after just getting in the raw genomic data.
So when you say raw genomic data, is that are you doing full genome sequencing for this comparison, or is it certain like genetic markers like single nucleotide polymorphisms? Like, because one of the things we've talked about in the past here on the show is it's hugely computationally intensive to do whole genomes, right? That's not even the cost anymore that's prohibitive. It's just like the processing power. Are you able to just bypass that and use the whole genome, or is it particular elements of the genome that you're going to look for in sequence?
Yeah, so there's a lot of different sequencing methodologies. And again, it depends kind of what you're looking for and what the primary question is. So a lot of the work we're doing is really focused on discovery. So we kind of want the most information that we can get. So we like to do deep whole genome sequencing when we can, but that still is costly. The costs have come way down, and they'll continue dropping. So then so you're trying to get not only the breadth of the entire genome, but also a deep enough coverage at any individual loci. And then there are cheaper alternatives where you can get more shallow depth, but then there's less. Obviously, the reads are sparser at any given location. So it's a balance. There's certain methods that are developed for lower coverage sequencing, but you can really do the most kind of powerful discovery with the deeper whole genome sequencing.
When you're going about this in the discovery methodology, right? How do you know when you've gotten something like a hit? Like this is in fact a candidate that I should pursue and use it as a marker for there being a potential tumor that's, being indicated by the liquid biopsy versus just being like, this is a false positive. Where is that line? Because like it's almost the so much data that I can have almost a misdirection. And how do you go through that? So like I don't know. We always find the computational side of things so cool here because you have to threshold things and you have to figure out what you're going to pursue and when you're going to pursue it, especially with that much data. Like it's either you have a treasure trove or you've got a map with a bajillion X's on it, and which one do you go for first, right?
Yeah, and I think that's kind of exactly what you said. That there are some thresholds that you need some level of kind of read support to be able to confidently call, whether it's a mutation or some type of structural variant or copy number alteration. So all these things that we kind of look for that are features about, potentially the tumor, to say, is this just maybe some type of biological noise, technical noise from the sequencing processes, or is this true signal? And so that's really more on the tool development side of things, and like the field has done a lot of work, and some of this comes even earlier in the phases of sequencing technologies developing even faster to try to eliminate some of these technical artifacts where you can get kind of false signals.
Nice. So that's when you can parse out if you have a mutation from a patient that's just variation from patient to patient. It has nothing to do with the cancer versus actual cancer indicating mutation.
Yeah, so there's also a large number of databases, kind of from all these population level studies. That there's just kind of population level variants that occur that are completely benign, and then there's also a lot of these databases that kind of encode likely pathogenic or likely benign, that don't have any function in terms of like coding implications or protein coding. So the standard is kind of central dogma is DNA, RNA, or protein. So there are a bunch of these kind of passenger mutations that can happen that are just completely harmless, and so we can easily just filter those out, and they're not going to have any functional consequences.
That's really cool that you've got almost gigantic databases at your disposal of past data that people have collected, and almost like you can build on top of that versus starting from scratch, which I feel like is really empowering for the method. So let's say you discover a hit. What does a hit look like? What does a hit, I guess, mean for you as the researcher? And what do you do with that information? What is your next step?
Yeah, I mean, so it depends on kind of the scope of work we're looking at, but depending on the different cancer contexts, there's certain driver mutations that we're interested in, key genes that are important for that cancer that can then impact what treatments might be available to that patient, or some that might they might have. We're looking for development of resistant mutations, or things that are impacting kind of amplification, leads to increased proliferation. This might be a patient that we should monitor more closely. So it really depends on kind of at what stage of disease we're looking at, and kind of how closely we're collaborating. A lot of the work we're trying to do, these are samples that are collected through trials, and we're kind of analyzing them post hoc. So looking for mechanisms of response and resistance kind of after the patients have undergone therapy. Unfortunately, in many of these cases, like the outcomes for these patients are already known, but hopefully we can use this information to inform future trials or indications. So like a current project that I've been working on is for a new prostate cancer therapy for late stage metastatic disease, where there really isn't many treatment options available in this space at all. And so one has recently been approved by the FDA. So the first round of patients have been put on this new therapy. But roughly half of the patients don't see any objective response. Some patients see tremendous response, but we really don't know why, still, kind of what those mechanisms of response and resistance are. And so in this particular trial, we're able to get liquid biopsy samples every time they came in for treatment, so every 6 weeks. And with that, we can monitor changes in kind of their genomics, their epigenetics, to look at not only like genotype changes, so looking at mutations, but then phenotype, like how is this tumor actually expressing and responding to therapy? Like what are the transcriptional changes? Things are being switched on and off, to then try to learn about how patients who are resisting or showing no objective response, what's really driving that. And so now that we've collected this information, this can then inform future trials or patients that are currently being treated with this drug. And then our clinical team at Fred Hutch has this information on hand. They're doing this liquid biopsy sampling, and they can use this readily in their clinical practice.
So, when you say late stage for these particular patients, what does that late stage mean? Is that the 4B equivalent where we're already like it's already spread throughout the entire body? What does that mean for those at home? Um, yeah, in terms of that late stage, like that, and that the prognosis is already like probably assumed to be bad.
Yeah, so in this particular setting, these patients have multiple metastatic sites. So they're prostate cancer patients, so it's spread beyond the prostate into multiple different sites. And prostate, the most common areas of metastases are to the bone. The liver is very common. So anytime it's then spread to the bone and other visceral organs, the outcomes are very bleak. And then these particular patients have had failed on many prior treatments, most of them. So this is kind of like one of the final last line options that's available. There are a few other chemotherapies, but many of these patients had failed on prior chemotherapies as well. prostate cancer is a cancer that if you catch it early, it can be very treatable, and patients can live a long time. But with many of these cancer types, once it starts to spread and gets to the metastatic state, things get very tricky. And then these tumors, they evolve quite rapidly, and they, like you said earlier at the start, they become very heterogeneous. It doesn't look like the primary tumor at all, and even different cell populations within a certain metastatic site might not look like each other. And so that's when it becomes very challenging to treat. These targeted therapies that are extremely powerful might not capture all of the tumor cells in that patient anymore, because they've changed so much over time.
So when you're looking at the different patients and these can map back the disease progression of the tumor, are you able to see the changes in almost gene architecture over time when you're taking these 6-week samples? And what does that look like? Naively, I would presume that it's just increasing mutation load in almost like a linear curve. But what does that look like in terms of like when you're in the data? Is there a way that you can actually map like, okay, this is late stage mutation that accumulates versus early stage mutation? You can actually stage the tumor based on this liquid biopsy. Like what info can you get from this and how can you process it?
Yeah, so this is very complex. So there's a lot of work kind of broadly in this space in like phylogenetics. So trying to map kind of that lineage, the different mutations that are shared between these different tumor cell populations. So they're called clones. So these different subclones develop kind of their own set of mutations. They'll share some of the original ones from kind of the primary kind of founder clone, and then as that spreads to different metastatic sites, they develop their own set of mutations. So computationally, you can kind of try to work back and figure out that lineage. And then also that gives you, and then layer that on with the different treatment pressures that have been applied to any given patient. And you can try to figure out like what the tumor has done to try to evolve to escape those pressures. And so I think that we'll keep it anchored in the prostate cancer space because that might be helpful. So prostate cancer, like one of the key targets we're looking at is the androgen receptor. And so a lot of these patients will undergo androgen deprivation, and the tumor will find ways to try to turn those programs back on. And so those are things that we're looking for, and you can kind of track over time and compare with different metastatic sites. It's pretty crazy the types of mutations and structural rearrangements the tumor will undergo to turn this machinery back on and basically go into overdrive, even though you've tried to basically chemically remove the ability for the androgen receptor to operate normally.
So is it fair to say that much like tumors are very unique per the patient, the direction in which they go in terms of mutation is also going to be unique to the path that that patient's taking insofar as the different treatments and the different things you might like, the different drugs that you're trying to hit against that tumor, and then how that cancer is going to try to circumvent that treatment?
Yeah, and so yeah, depending on how a patient presents, like they could be good candidates for a certain subset of therapies, and then you apply that treatment pressure, and it might work initially, and then a cancer will figure out like different reversion mutations or ways to try to counteract that treatment pressure you're applying. So there are, like I said, depending on the cancer type, there are known drivers that we're looking at, but there is a lot of heterogeneity in the patient populations. And even though like we might look for some of those, and that has a strong signal population level overall, the individual patient might not fit cleanly on within the normal statistics that you see in the trial. So that's why it's really important to try to tailor as much as possible. Like, this is really a precision oncology, to use the buzzword type of aim, and that we can really be very specific, to identify within this specific patient how they're responding to therapy and kind of what's changing in real time. Really, the only limitation is the sequencing costs and the data analysis. The actual impact on the patient, like they're getting their blood drawn anyways to do their routine work. It's nothing more than that on their end. So it's more kind of the overhead. But we can get that information in real time, which is very impactful.
Can you give us an idea, Patrick, of just the cost of one of these sequencing experiments? Like for one patient, roughly, how much do you say that costs? Not I mean, I know your time in terms of like the bioinformatician behind it is also a pretty large chunk, or not just you, but like anyone who's going to be analyzing this. What is the cost of the sequencing data alone to try to get this per patient? Just to give folks an idea of while the costs have come down, this is still a pretty pricey endeavor.
Yeah, so for low pass, so again, that's very shallow coverage. So for what we can do with that type of data is be able to say what percentage of, or what percentage of this cell-free DNA that we detect in this blood sample is coming from tumor, right? And that's incredibly powerful. If we detect very low levels of circulating tumor DNA, like that's a very, very good sign. But once you start to get into detectable, or sometimes these are very high levels in patients, that's a bad sign. It's been shown across cancer types, so that's very prognostic for future outcome is the level of circulating tumor DNA. But you don't have the ability there to really look at mutations because it's such low sequencing depth. We just don't have the confidence to make those calls. So for that, it's a couple $100, so 1 to 200 usually. And there's a bunch of different platforms. It's pretty quick, routine, and the tools and the readouts of that are pretty gold standard these days. So that's usually like the first pass we do to triage. If you want to go deeper in there for deep whole genomes, those can be $1,000 to $2,000 per sample, and different targeted panels that will also get you really strong depth at more focused locations for those driver mutations that we might be looking at, is kind of going to be in that similar range of $1,000 to $2,000.
So we could be looking at the end of the day, 5K per patient per time point, possibly, right? Because it's not just a one-off $2,000 for that deep sequencing. It's like you were saying, every 6 weeks for that particular patient that you were collecting for. Is the end goal to be, I can just take a blood sample from a prospective patient who might have cancer or maybe has already been ID'd and kind of gauge where they are along the line of the disease progression? Is that the big overarching goal?
Yeah, I mean, so there's a lot of interest from pharmaceutical companies in this space to build these multiple cancer early detection tests, and then you can just give these out to anyone, and anyone can send in their blood, and it can tell you if you're positive for cancer X, Y, and Z. But for us, we think about this much more as a way to kind of understand how the cancer is responding to treatment pressures, and then so that it would be looking at patients over their treatment time course. But just like to put things into perspective in terms of the costs, the the costs of the therapies themselves, let alone the hospital costs and all that's associated with that, this is minuscule compared to that. A single dose of this prostate cancer therapy I was talking about, the drug and associated costs is upwards of $80,000 per dose. So just a little bit more on top, we can get a lot more information and say, is this even an effective use of $80,000, or would this patient be much better served by a potentially much more effective therapy for them?
So, in your goal, this is being developed for those that have already been diagnosed as a diagnostic driver for that personalized treatment of this is going to be the best path forward given your genetic mutation load almost. Like this will be what the tumor is responsive to. This is the best course of action to help you survive. So that's what we should try to do first. Is that a fair assessment of your goal on this front?
Yeah, absolutely. I see it very much as a dynamic readout of how that individual patient's tumor is responding to therapy or not responding.
So, what all goes, Patrick, into when you're developing this pipeline, right, for analyzing the data coming in? Because there's this goal of helping patients, are you also like almost on a video game dev side of things where you have to think about UI and user interface for how you're developing the program? Or what all, because it seems just so multimodal of what you're having to build. What all goes into building this kind of software that is going to be able to analyze this data for anyone just even outside of your hands?
Yeah, so a large part of what we do is, like obviously we want these tools to be used by us, like within our lab and the folks at Fred Hutch. But these are hopefully publicly available tools that can be used by the wider scientific community and hopefully impacting patient lives far beyond our center and making real contributions to science kind of beyond our small little slice. And so with that, it has to be something that other researchers can easily pick up and use. So that comes with like all these tools are available on GitHub, and hopefully they have clean readmes and they're user friendly and they give you interpretable results. But that can mean many different things depending on your background and where you're from. So generally, we're developing things for other kind of computational researchers or people in kind of the cancer genomics space. But occasionally we do develop tools that are, in concert with clinicians. And so that it can be helpful to have kind of a nice little web page where they can input a few different metrics, and then it, gives them a nice little like risk score or something of the like. A lot of people kind of want to know at the end of the day, what is my risk of X, Y, and Z? And so anytime that you can kind of develop some type of prediction score for that, it's very clinically useful and informative. And so, we try to think about things that way as well, not just, obviously the science is important and we want to make sure that the methodology is sound, but then also thinking about like, what is the clinical question we're trying to answer? And then how can we develop the tools and kind of the wrapper around that to make it useful to the broader community.
Yeah, I feel like it's a two-pronged difficulty, right? Is developing the original program to ensure that it works and it's hitting the goals that you're trying to do, but then it's also adaptability and how likely it is that folks are going to adopt it. Because it's like you don't want to just like invent a life-saving technique that no one's going to use, right? So it feels like that could also be a difficult side of things, and it also goes to show how many different hats you're wearing as like the researcher here, right? Like I feel like it's far more than just the I'm taking blood samples and running them through a sequencer. It's like building the pipeline of what's going to be detecting of what's important, making the correlations between patients, looking at outcomes, and also trying to get people to adopt this methodology so making it the easiest possible way to use it. And then like chat says too, like documentation of like from the dev perspective, which no one enjoys doing, but then having to put that in as well.
Exactly.
So
so a lot of what we do, like it's going to be published in an academic journal, and so that has a big piece. Like you want to make pretty figures and like nice infographics, so it's easy to understand kind of the data inputs at a high level, kind of the methodological structure, and then what your expected outputs, and then hopefully you're showing meaningful results and in patient data, and then some of that's kind of like simulated data depending on the scope of the work. And then that will hopefully drive people to use your tool. And then there's, the hard thing is the documentation and making really clean, like for those that are familiar, like really clean readmes and like helper outputs and things like that. So people can debug errors that are bound to come up. I mean, to have everyone's data that's coming in fresh, perfectly aligned with your tool it's a near impossible task. So having kind of those steps to try to get out in front of that is very hard.
So what does your day-to-day look like? I guess one thing that always is wanting to be demystified is what does a scientist do? And we've talked about these really almost gigantic pillars that you're building, right? Of like each of these elements that needs to come together to be able to make this potentially life-saving tech. What does it look like of what you're doing on that day-to-day that's helping you strive towards this goal?
Not directly answering your question, but I usually bookend my day with something physically active, just to kind of get my body and my mind going, and so get things going in the morning, and then kind of settle down in the evening. But it turns out to be great thinking time. Occasionally, there's some times where it's out on a run or a bike ride that the brain starts thinking about whatever the previous days or weeks questions were. But I think, one of the things that's really, exciting about science is that it's something that like the part of your brain just never turns off. And there's always interesting questions to explore. So whether it's, reading journal articles or, talking with different collaborators, that part of your mind is always going, whether you like it or not. But then, in the office, we have a very kind of collaborative environment. So like I said, computational biology, that can mean a lot of different things. And so there are people in our office that wear a lot of different hats and come from different backgrounds. And one of my closest collaborators, a fellow postdoc in the lab, his background is in physics, and my background's in statistics. And then we're meeting here in a computational biology lab doing cancer research. So a lot of that is, we're on our computers most days. We're not a wet lab, so we're not actually touching the patient samples or doing any of that extraction. So it's very much head down in the laptop, doing its heavy coding, and then meeting with different project teams. And we work with a lot of physician scientists, that are actually seeing patients in the clinic and then run research labs themselves. And then obviously we work with a lot of kind of more basic scientists who are the ones that are actually processing the samples and doing the more experimental work, whether it be kind of more mouse models or other different types of basic science that I personally don't have the experience in, but it's very important to work with those folks and have those good relationships.
So it seems like it's extremely multimodal, right? And like multidisciplinary of what you're trying to study and how you're studying it. How do you manage all the different data sets, right? Like you're just saying you're talking with a physicist and then a data scientist and a clinician scientist. Like, are you the hub? Essentially, it sounds like to me that you probably have to be the hub, right? And everything has to be coming in towards you, of you deciding what kind of data that you need. Is that a fair assessment, Patrick? Like, I'm seeing you almost as like if you've seen Spider-Man, you're the dude in the chair.
No, the data management piece is very challenging, and that's we have a lab manager that helps try to corral the different sources, especially when we have so many different projects and collaborations across the hutch, and then we have external collaborators as well. These are massive data sets, and the network just keeps growing. So yeah, it is quite a challenge. We try to have structured documentation on these things, but these are living data sets. Things get added all the time. So you really have to be on top of kind of that project management, and knowing kind of where your data lives, and making sure it's backed up correctly. We have things on the cloud, things kind of on our cluster environment, and then managing all the different project folders. So yeah, I think it is a tough task.
What is your ideal data finding? So like if I were to tell you that I'm going to get rid of for you all the variables, I'm going to give you all the money you need for this project, what is your ideal data result? Like what is it that, if again I can guarantee that everything's going to work perfectly, what would you want to see? Like as in like this was successful, this is now going to make a difference, versus what do you end up seeing? Like so ideal compared to reality.
Yeah, I mean, so with cancer data in particular, like what we're really focused on is patient outcomes. So there's overall survival, like that we want to see patients living for as long as possible, and that's going to be the gold standard. Whether you're looking at it at a regulatory level, like that's what the FDA is grading all these drug companies on, is are they making meaningful impacts on patient lives? And then also, within that, there's kind of different response metrics. Like, are we at least, maybe they might not be surviving longer, but are we delaying the time to what they progress or their cancer recurs? And so that's kind of, what I hope to see when we run some type of analysis. Maybe if it's, looking at a particular pathway or something we think has meaning, and then you relate that to outcome, and then hopefully you see that it does have an impact, and then that's your major finding. Hopefully it's something you can target or something you can track, and that's having a direct impact on how we're managing patients.
Have you gotten anything where it's been really surprising, where it's like there's a pathway or a gene where you're like, what is this? This wasn't something that I even thought about. But then, in hindsight, it's like, oh, okay, I can see how this kind of can make sense. Again, going from that post hoc look, it's kind of a little bit easier to do like that, but still there could be surprises. Like, when you get something like that, like, is there an example you can give us, and what was kind of your role of validating something like it, where you're like, okay, now I'm convinced that it's not just an offshoot, it's a real thing?
Sure. Yeah, I'll go back to the prostate cancer space. So this drug we're looking at, it targets it's a targeted therapy that attaches to this It's called PSMA. It's expressed only in prostate cancer cells, pretty much exclusively. And then it attaches to these PSMA expressing cells and releases a radiation dose, hopefully killing those cancer cells. Um, One of the mechanisms that relies on kind of these AR dependent prostate cancer cells to express the cell surface target. There are prostate cancer cells that will transition to a different kind of phenotype called neuroendocrine, and so these don't express that cell surface target that this drug can bind to. So one of the things that we thought we would see was that kind of over time, these patients that weren't responding to this new therapy would be losing that cell surface target, and in turn, kind of changing their phenotype, and finding ways to become AR independent and more neuroendocrine like. It turns out we didn't actually see that, which was a bit surprising. And instead, that most of these patients who were still progressing, their disease was progressing on this new therapy, still expressed the cell surface target, meaning that there were lesions that this drug could bind to and could still release its radiation dose, but it still was not being effective. And we didn't really see strong signs that these patients were transitioning to a different phenotype. Instead, they were had this much more kind of proliferative, aggressive type phenotype that was basically their cancer was more aggressive than the drug could combat, and so that was something that we saw that was really stark, and kind of was against our original hypotheses and what kind of many in the field thought would be the primary mechanism of resistance. It was actually not the case. So I think there's maybe that will still bear out as more studies evolve, but there really wasn't any strong signals there, which was quite surprising.
Yeah, it's really interesting that all of a sudden it just started growing better, like faster than you can slough them off. And presumably it's not something where you can just dose up the drug, right? Because eventually it's already poisoning the patient, right? At a certain point, you're going to be causing more hurt than good. And so then you have to find some totally other kind of treatment source.
Exactly.
Yeah, that's fascinating, but also really frustrating because it would have been so much cooler to find, like, oh, it was just this other target now that we can develop a drug to, and there we go. But when it's that speed, I guess the answer is probably right, just multiple treatments at the same time to try to counteract the growth efficiency of the tumor. But yeah, that's a really tough one.
Yeah, and so that's, I think, one of the really cool things that we can do with liquid biopsy is our lab has developed methods that we can kind of detect the expression levels of these different cell surface targets. So we have to infer that. We can't directly assess that from the DNA, but we can develop tools that allow us to say, is this actually going to be expressed by these tumor cells? And so we can see that these tumor cells are still expressing this primary target, but they're also expressing other cell surface targets that there are drugs for, or we know that drugs are being developed for in prostate cancer, or across cancer contexts. We can do this too. So that's also offers hope and opportunity that these patients could have, or may already have, other drugs available to them, even though this one they don't seem to be responding to.
And the beauty of this work, right, is that you can, in theory, use this for any cancer type. So while you're developing it in prostate cancer, if someone were to develop the same pipeline in terms of collecting the data, right, then in theory, the pipeline you develop, you just apply it to that data set, you figure out genetic markers or epigenetic markers that are associated with disease progression, and then use it to, again, stage what treatment that particular patient can need for that cancer type. Is that correct?
Yeah, absolutely. So almost all of our methods are kind of cancer type agnostic, and can be applied across the cancer care spectrum. And that's really what we strive to do. Obviously, we have very close collaborations, and projects are focused in a specific area where we collect samples. But what we're really looking to do is to make these tools as widely used as possible.
And again, prostate cancer is what you're focusing on, just because there's really no treatments available, or like very low efficacy treatments available for these patients.
Yeah, so Fred Hutch in particular has a phenomenal prostate cancer program. So that we have, our lab in particular has some strong collaborations there, with some of the clinician scientists that are really involved in seeing the patients and then thinking about the research. And this particular project I'm talking about here, again, is for this late stage disease where it is a new therapy that we don't know much about, and it is a kind of the only one of the only options for this patient population.
A question from the chat is,
Sure.
for this methodology, is it only on tumor-based cancers, right? Because of you're doing the liquid biopsy, or can this also work on blood cancers? Like, what cancer, if any, are you limited on in terms of type?
Yeah, absolutely. So a lot of this work like is has been used widely in the liquid tumor space, so the blood based cancers for a long time. I think that is one of the tricky things is you're getting a normal blood draw, so you're getting kind of the blood component as well, not just the circulating tumor DNA from solid tissue. And so when there can be some confounding there that depending on the individual patient, that has to be taken into account.
Speaking of confounding, another question from the chat too is, given that these are all like, the tumors are trying to proliferate, right? These can involve different treatments, especially for the different types of tumors. How is it that you can cross apply the research, right? So like if I'm studying, cancer X and you're studying cancer Y, and the treatments are also different between the two types of cancer, how can we utilize this method to ask questions about both? So essentially the question I think is coming down to, these are different types of cancers, the treatments are different. What's the logical argument as to why the methodology is going to help inform treatment for either type patient?
Sure. Yeah, no, that's a great question. And we're talking kind of like very abstractly and at a high level, but so there's a lot of different tools that we have developed kind of in our lab, and then other researchers in the field have developed. And so a lot of this is looking at gathering different pieces of information kind of about the tumor. So we talked about there are tools developed to call mutations specifically. There are tools developed to call different types of structural variants or copy number alterations, these things that are kind of hallmarks of cancer, regardless of cancer type. And then we also have different tools to infer phenotype or gene expression changes that are very important to kind of understand the cancer phenotype, not only its genotype, its genetic makeup. And so this can be applied kind of across disease contexts. So it takes kind of gathering all the data, running the various tools, and then where we come in as researchers is really trying to integrate that information into your specific disease context, where you're then thinking about kind of the standing research in that field, or your current patient you're looking at, your patient population you're looking at, and thinking, okay, what of this is important? How do I synthesize this information and make meaning out of it in this specific context?
And this is why I love the questions from the chat too, right? Is how do the kinetics of the tumor and the cancer work into this? So, for example, even from the chat, some tumors progress faster than others, right? Just because of accessibility to nutrients, accessibility to being mutated. So, how are the kinetics playing into this? Because like one type of tumor, you're taking samples every 6 weeks and being informed, but other types might have to be different in terms of timing, or is this all just like, yes, we know we're kind of frontlining it and trying to figure that out per cancer type?
No, this is a fabulous question, and it's something that we don't really have solid answers for. We have some information, but it's like an active area of research and some heavy mathematical modeling. Some physicists also love this work too. It's a lot of dynamical systems work. But so the rate at which you detect circulating tumor DNA is very much going to depend on disease stage. So later stage patients are going to have much more circulating tumor DNA present because they have more total tumor volume to be shedding into the bloodstream. So patients, hopefully, say they underwent surgery and removed kind of a local lesion, they should see almost no circulating tumor DNA. And that is like a great sign post-surgery that was successful. And then you can monitor that over time, and you might see slow upticks if there is kind of recurrence of that disease. So that's one thing, just kind of total tumor volume, depending on the number of different kind of metastatic lesions a patient may have, or if it's just kind of a localized setting. Also, different sites can shed more into the bloodstream than others. So your liver processes a lot of blood. So anytime there's a tumor involved in the liver, those are going to be much higher shedding. whereas something like the bone is a much lower shedding tumor. So there could be many bone lesions that you can detect on scan, but you might not be able to detect that as readily through ctDNA. So these are all different types of considerations. And then so a clinician, when they're getting this information, they're looking at scans, they're looking at other lab values depending on the different cancer type that may give them information. And then if we can also layer on additional information from circulating tumor DNA, whether it's just like a quantitation or maybe something about, like when we're talking about different types of genotypic information or phenotypic information, this just gives clinicians more tools to be able to better adapt their treatment regimens for their patients.
That's really, really cool. Like it's a very robust program that you're developing, right? If every question that we're asking is like, oh yeah, we have a method for that, we method for that. And I like too that even in the answer, it's like it's very multimodal, right? It's an informative tool for the physician in addition to everything else that they have access to, right? It's not necessarily replacing anything, it's just in conjunction with, and I think that makes it all the more powerful.
Exactly.
You mentioned, Patrick, mathematical modeling. Now, is this something that you're also doing as like an element of the project? And if so, could you delve into it? Because I always find mathematical modeling, like in it from an ecology perspective, to be really fascinating of like how populations and neurons even behave. But how does that, if you do in fact too, integrate into cancer in this question that you're asking?
Yeah, this is really interesting. I feel like you would love to be a fly on the wall of our postdoc office. So like I'm in statistics, so I think about things in terms of like distributions. So I don't know how familiar people are with that. I mean, like you have your normal distribution, your standard bell curve. Usually, people are familiar with that. And there's also kind of this theory of like a Bayes theory. So you think about kind of your prior information, and then you have some distributional assumptions, and you gather new data, and then you have your posterior distribution. You continually updating your information as you move along. So that's how like my brain works in terms of when we're thinking about tool development. And then a physicist sees things from more the dynamical systems perspective, and kind of like information entropy and modeling things in that sense. And so there's sometimes a good convergence there, but it just there's different frameworks of thinking about kind of how we want to model the data we're looking at. So ultimately, what we're getting is sequencing data, which is just a pile up of reads, and then their kind of their the read sequence, and then thinking about the information we want to glean from there and how we can best kind of model that and get to that end goal. And there's no right answers. People are constantly developing new methodologies, and this moves as the field moves. And I think that's kind of the fun part, is playing around with these different modeling assumptions, thinking about what different levers we can pull. And we haven't even got into AI, but that's a huge piece of it and has really accelerated the pace at which we can iterate. Yeah, so I think that's the fun bit, is thinking like there are no right answers, and it's all discovery and kind of playing with the data, and thinking about how we can make meaning out of it.
As we wrap up, Patrick, first of all, thank you just so much for your time and your expertise. This hour has absolutely flown by. It was a lot of fun learning and just chatting about what you're working on. What are you most looking forward to? Like, if is there a particular experiment or milestone that you feel like you're coming up on that's going to make like a really big, that you feel like is going to make a really big difference that you're very excited for?
Yeah, so like, hopefully we are moving towards some publications soon coming out from our lab. We have some projects wrapping up that are hopefully going to be submitted. So that's like, it's a big milestone in terms of like the main body of work, but then also the start of another long journey. The publication process in academia takes a very long time, which is a bit unfortunate because I think it slows the process of getting science out. But these are still things that regardless of what that looks like, we can start implementing, and we have implemented kind of in the center, and really making impacts on how we're thinking about treating patients. Another thing I'm really excited about, and maybe you'll get into this with Helen, is Obliteride is a super fun event that Fred Hutch puts on, another fundraising event, a charity bike ride and run walk. That's just it's a great atmosphere for us coming together as a cancer research community, patients, survivors, friends and family. It's just a really fun weekend. So that's coming up in August. So really looking forward to doing that. And it's always great to ground the work we do on a day-to-day by interacting with patients and getting to talk with them about some of our research and the work we're doing. So it's always fun.
That's awesome. Well, Patrick, seriously, thank you so very much for your time. Good luck on the paper. I know from too many experiences how frustrating that can be, but I feel like you got this. This is really meaningful work, and we really appreciate you sharing this, sharing your knowledge with us, and just working on what you're doing. It's important stuff. Is there anything you'd like to leave the community with? Any final message? Even help plug your socials, whatever you want. How people can support you? What should people do to support you and your goals and your aspirations? The floor is yours.
Yeah, first of all, I really appreciate the opportunity to come on here and the work you're doing in supporting Fred Hutch and the community's doing in supporting Fred Hutch. I heard you mentioning this when I jumped on that like every little bit does help. I know we talked about some numbers of how much these things cost, but you'd be surprised. And so going back to Obliteride, some money that our team raised directly went to sequencing costs, and that led to an additional, I think, 10 or 12 samples that really like rounded out a really strong analysis and had a big impact on a bladder cancer project. And so those are all just like small dollar donations from like our friends and family, and then our broader networks. So just I think any little bit helps kind of keep talking about the science and the work we're doing. It's a very toxic research environment right now, unfortunately. So I think having events like this, talking with your friends and family about how important cancer research is, definitely helps. So thank you to everyone who listened in, and hopefully we can keep the chat going.
Well, we appreciate you, and whenever you want to come back, Patrick, the door's open. Truly, thank you so very much for everything you do.
Science is for everyone, all backgrounds,
Thanks,
all
everyone.
ages, all
Thank you.
genders,
Have a
and all
good night.
walks of life deserve an education. Come be one of us.